Swayam Prabha. Digital India. Educated India.
So, today we continue with the cell signaling. Remember in the last class we began with this RTK. So, we primarily learnt about how the RAS GTPase GDP GTP exchange helps in fine tuning the signal, right. Only as long as the signal comes you will have the RAS GTP, otherwise it is automatically becoming RAS GDP and inactive because of the actions of gap and the intrinsic hydrolysis activity, right. So, we continue to other signalings. The next one is a small variation on RTK. Oh, okay, the RTK itself, one more example. So, this is this is just to illustrate that the RTK pathway ligands are diverse. So, here for example stem cell factor, when it binds through a receptor tyrosine kinase receptor, like in the case of melanocytes. So, the receptor here is the KIT, which we have already seen as an example earlier, right, and MITF also we have seen as an example stereotype of a transcription factor, right. So, here you see the signal. So, you find the red and green. So, the color has not come out really as red and green. So, what you are seeing here is actually you see at least white shade around the red. So, the red is actually one protein and the green is the other one, that is MITF and KIT, and when they both are there then they look whitish, supposed to be red and green, and the overlap should be yellow. And you see that in these cells, these are melanocytes, okay, and in KIT knockout you do not see that. So, the migrating melanocytes express this, and that is required for specification of the melanoblasts that are eventually going to become the pigment producing cells. And that is why you have skin color aberrations. Remember that 4 log of the mother and daughter in that MITF mutant. Okay. So, a small variation of that same pathway, again this is a similar receptor, but it is called Janus kinase. The difference here is the way the signal is transduced. So, the transducer is STAT and this protein is transducer as well as that goes into the cytoplasm, nucleus to activate transcription. So, like here the signal transduction happens via a cascade of proteins involving finally a couple of phosphorylation based regulation, and then a phosphorylated protein gets into the nucleus, right. So, the transducer, if you take this adapter protein or GNRP, they are not the final effectors, they are not the ones that go inside. While in JAK-STAT pathway, that molecule is directly, the transducer is the one that directly gets into the nucleus and activates, and as a result it is called signal transducer and activator of transcription. STAT stands for signal transduction and activator of transcription, AT for activator of transcription, and they dimerize and go inside and activate downstream signaling. So, this sort of regulation works in the example of casein gene in during milk production. Prolactin binds to the receptor and then that leads to STAT activation. So, here is an example of a mutant condition. So, here this mutation in this extracellular domain of this FGF. So, remember this entire thing is called FGF, right, fibroblast growth factor family. In that we saw RTK, and now we are seeing STAT as a small variation on the same group. So, when you have a mutation like this kinase domain remains constitutively active even when the signaling is not there. So, the receptor is constitutively active, and as a result these cells, these chondrocytes prematurely differentiate without making enough of the starting material. Due to that, the bones become short and you get this what is called thanatophoric dysplasia condition. Okay. So, this kind of highlights the importance of JAK-STAT pathway in limb development, and this is lethal. Okay. So, this lungs can't expand and breathing is not possible. So, this will be dead on arrival. Okay. So, it's not going to be viable. Okay. So, the next one is hedgehog pathway. Okay. So, each pathway has unique characteristics in terms of how the signal is transduced and what kind of molecules are involved. So, therefore, it is not that hard to remember them, and there are only 4 pathways. So, the uniqueness about hedgehog is it is a protein signal that gets cleaved, okay, and only the N terminal portion is secreted out, and that N terminal portion is cholesterol modified. So, you have encountered many post transit modifications, particularly phosphorylation, methylation, acetylation, these things you have already seen, right. The last 2 I said are in histones. So, this is an example where cholesterol modification happens. I do not know any other example where cholesterol modification happens, but there are other lipid modifications are there, prenylation, palmitoylation. So, those kind of modifications are there. Sugar modifications are there, ADP ribosylation happens. But this is one unique case where there is cholesterol modification. So, this cholesterol modified N terminal portion of the protein is the signal here. So, that is what is the hedgehog. So, this word hedgehog is based on the Drosophila phenotype, okay, where this was originally found, and in mouse the ortholog is called a sonic hedgehog. So, this is a whole mount embryo in situ showing expressions. 3 tissues are specifically marked. So, you have in the nervous system and you have in the limbs, and then this is the gut region. Okay. So, 3 arrows showing where they are expressed. So, these are usually important for tissue boundary making. Okay. So, boundaries are defined primarily using this signaling. It is required for limb development and neural differentiation as well. Okay. So, involved in somite differentiation, to make the cartilage out of dorsal part of the somites. So, the ventral, from the ventral part, dorsal will be by the next signaling vint. So, let us look at how this pathway is. So, I will tell you something which will be catchy and then we will come to the pathway. Yeah, look at this picture, right. This is called a cyclop, not a Greek mythology, real life. So, there are some plants that grow in some parts of the world like in Arizona, California, etc., and they produce certain molecules called an alkaloid called gervin or opamines like cyclopamine, and if the sheep eats this and when her offspring comes out, it will have problem in development. So, these alkaloids, they inhibit cholesterol biosynthesis. As a result, enough cholesterol is not produced during the embryonic development, and due to that you have craniofacial development problem. The brain, both the 3 parts of the brain do not form the separation and they form a fused structure. Due to that you develop one eye, and due to that it is called cyclop. Okay. So, this illustrates the developmental importance of cholesterol. So, if anyone says cholesterol is bad, you remind them you would not be alive without cholesterol, okay, simply because you eat the wrong food and wrong quantity at the wrong time and do not do physical exercise. Do not blame the molecules. Now that you have seen the consequence, you will be very much interested in how the pathway works. This pathway again works like the accelerator pedal in car, there is negative regulation. So normally you will have this cubitus interruptus. This is attached to microtubules via these cos2 and fused, and when it is like that, this pKa and slim, they would cleave this only when it is attached to microtubule via these two proteins, and the cleaved part of Ci goes inside and acts as a repressor. This is what normally it does. And whenever you have Hedgehog binding to this transmembrane receptor called patched, which otherwise keeps the other membrane bound protein in a repressed condition, okay, smoothened. So these are again based on the Drosophila embryonic phenotype. So when Hedgehog binds, this inhibition is released, and due to that the smoothen inhibits these two proteins. So people still do not know clearly the molecular mechanisms involved in it. So the end result is these two are inhibited, one. Second, that smoothened by phosphorylating these two proteins, cos2 and fuse2, releases Ci from the microtubules, and now the Ci is not cleaved because these two are suppressed. It translocates and then it recruits a protein called CBP and it becomes an activator. It is no longer a repressor, instead it becomes an activator. So this is how the Hedgehog response genes are activated. Okay, this is how this pathway works. So here the uniqueness is these molecules, these are specific to this pathway, and then you have this cholesterol modification. So that is how you can recollect this and will not get confused with the other pathways. Alright, so the next one is Wnt signaling. So Wnt, actually the word comes from wingless, which is a phenotype of a Drosophila mutant, and integrated, which is a vertebrate ortholog. So both together it is called Wnt. I think the vertebrate ortholog was first found in Xenopus. So these are glycoproteins that are rich in cysteine. So they are important for the dorsal somates to become muscle. So the ventral one become cartilage through the action of Hedgehog, and the dorsal ones become muscle through the action of Wnt, and it is required for specification of midbrain, again limb development. So this is again involved in limb development like the other ones, like JAK-STAT we saw. Then proliferation of stem cells in certain contexts, and more dramatically for the urogenital system. Okay. It is also required for female sex determination. So we will look at how this works. So this actually, it is a family of proteins. There are several molecules that are Wnt, and similarly there are several molecules that are its receptor, frizzled receptor, and which one of the member activates depends on which one is expressed in a given tissue. Okay. There are several Wnt like molecules, similarly there are several frizzled like receptors. So which one is going to be the ligand, it depends on which ligand is expressed in a tissue where a given receptor family member is expressed. That is one. And second, due to that there is divergence, like you have three different modes of Wnt signaling. We will see that, okay, 2 slides later. So before that, again like that cyclop, because this will catch your attention. So this is again an embryonic kidney testis part that is stained in situ hybridization for the Wnt4 RNA, and then you find it this purple color is the expression pattern, it is expressed in the kidney rudiment strongly. And if you do a knockout of Wnt4, so these are the wild type age matched same magnification kidneys, adrenal gland on the top, gonad at the bottom, and so on. And in the knockout, adrenal gland is intact, gonad is intact, but the kidney has become too tiny. So kidney does not form, so the nephrons do not develop. So Wnt signaling is required for this. So there are 3 different ways by which Wnt works. The first one was the first to be described, and as a result this is called the typical. In the western world they often call canonical, meaning which is like the textbook classic example. So many of the students in India do not immediately understand canonical, because that word comes from church based, canon means something that is said and there, and you are doing anything other than that is non-canonical. So the first defined pathway therefore is called the canonical, and the ones discovered later are called non-canonical. So the first discovered one has this sequence where you have this metabolic enzyme, glycogen synthase kinase. That one usually is associated with APC, which usually targets it for degradation. This is a tumor suppressor, okay, involved in colorectal cancer. And beta catenin you already know. Where did we encounter beta catenin? Cadaverine. Okay, cadaverine to actin connection is via catenins, right? So it's the same catenin. So all of them have other roles to play. Okay, so this is one function here. So I am trying to talk about that is just to highlight the point that these molecules are not dedicated doing only this one job, and therefore the dynamics of their concentration will depend on many factors in the cell, okay, not their role in Wnt signaling or their role in glycogen synthesis or regulating cell adhesion by modifying actin and cadaverine dynamics. So anyway, so this GSK3 APC, they target beta catenin for degradation, and as a result you do not have beta catenin going in and activating anything inside. But when you have Wnt binding to the frizzled receptor, again transmembrane receptor, and the transducer disheveled gets activated, that is going to suppress the GSK3, and now beta catenin is not going to be cleaved, and it will migrate into the nucleus and associate with this factor and activate transcription. Okay, so that is how this works. So again normal is negative. So this is just to fine tune. Only when the signal is there you are going to have that activation, and the moment signal is off, it is off. There is no comet like halo effect here, there is no trailing thing. And a small variation of this happens at the disheveled level. If the signal is transduced by a disheveled protein that is tethered to the plasma membrane via this prickle protein, then it is going to activate this Rho GTPase. We have learnt about Rho GTPase, right? Where? In morphogenesis, right? So ventral furrow formation, neural tube, the neural cells to invaginate inside. For all of that I said modifying actin by the small molecules like GTPase, that is the Rho GTPase. So Rho GTPase, similarly another one RAC, that is also small GTPase. So when these are activated, they are going to alter the cytoskeleton. So directly without gene expression change, you will have changes to the cytoskeleton when it is transduced this way. In addition, they do via JNK control gene expression as well. And a third version is branching even earlier than disheveled, so to an unknown protein which transduces, that is going to activate a phospholipase C, and that is going to cause calcium release from ER, and that is going to have transcription activation based on calcium mediated signal transduction. Okay. So these are three different versions by which Wnt signaling works. Okay, the next one is a mega family. Okay, it's a super family having multiple families inside. This is called TGF beta. Okay, And so here the ligand is actually the carboxy terminal, that is the mature form. Okay, the carboxy terminal is cleaved and secreted out, that is what is the TGF beta, and that dimerizes and then going to bind to the ligand, sorry, receptor. So it is required for extracellular matrix. Very soon you will see the importance of extracellular matrix in development. Then branching of epithelia to form ducts of kidneys, lungs, salivary glands, like wherever you need to have epithelium to tube formation. It is required for bone development. One of the family bone morphogenetic factor, this BMP, here you see this, and they were originally discovered for their ability to promote bone formation. Okay. This DPP, one of the BMP member in Drosophila, is required for germline stem cell maintenance. Without that they will not remain as stem cells, instead they will enter into meiosis. Okay. So there will be no stem cells without that. They are required for cell division, apoptosis regulation in many vertebral contexts, cell migration as well. So like that they are involved in many different processes. So they work in this manner. So you have the ligand, here we are looking at two different ligands, activin binding to it. So you have type 2 and type 1, two kinds of receptors, they dimerize when they bind to the ligand, and the type 2 autophosphorylates and then phosphorylates type 1, and that becomes the active version. And that active version is transduced by the phosphorylation of SMAD proteins. So SMA for small worm, okay, the worms are about half the size of the normal ones, they are really cute and nice. Okay, they are thin and small, so they are not like dumpy. So small, and MAD, I think MAD is I forgot the name, maybe Mothers Against Drunk Drivers or whatever. So that is in Drosophila, so they name crazy names. Okay, so both combined is SMAD. Okay, so it is SMA and MAD, common SMAD. So in vertebrates they simply call SMADs. So SMAD2, SMAD5, SMAD1 and so on. So if it is activin, it is SMAD2, 3 that gets phosphorylated, that associates with SMAD4, and that complex goes in and activates transcription. Instead if it is BMP, it is going to be SMAD1 and 5 instead of 2 and 3, that is the only difference, and then that is going to associate with SMAD4 and go and activate transcription. So this is how TGF beta works. Okay, so it is a family of families. Okay, so therefore it is a super family. So you have a lot of members, and they are really critical molecules in almost all organismal developments. Okay, so the last one, which is not those 4 main pathways, but this is again critical pathway, this is called apoptosis. Okay. So it is quite simple pathway. So already you know the story that in cell lineage tracing, I told you in C. elegans you can trace the entire lineage from starting from P0, and in that process people discovered 131 cells die always. So 959 plus 131 is how many? Whatever that number is what is produced normally. In that 131 die for sure, and the remaining become the normal worm. And this invariant lineage tracing of those 131 is what convinced people that there must be an intrinsic developmental program that makes a cell to die. So now we actually know in many organisms all cells are actually default is to die. There is somebody who has to tell, no, you are not going to die, and that is how they live. Okay. So that is how it works. For example, here if SED9 is not holding on to SED4, okay, it is going to dimerize and become active and associate with this protease okay, set 3, who is the executioner, they call it executioner in that field, cell death in C. elegans. Yeah. So it is going to be cleaved and active protease, and this is going to chew up all the proteins and DNA and kill that cell. Okay, that is what will happen if SED9 is not holding SED4. So in the normal situation, a cell that is not supposed to die, their SED9 holds on to SED4, and any time you get this signal Eagle1, meaning egg laying defective. Okay. So that is going to compete with SED4 to bind SED9, therefore SED4 is released and that goes on to activate SED3. So this is how it looks. In C. elegans it is easy to find apoptotic cells. These 2 images were taken by me, by the way. This is a very early larval germ line. You have 2 rows of germ cells. This is how normal nuclei look like, and when they are going to commit suicide, they become button shaped like this. Okay, and this is very distinct morphology. You do not need to come from Bob Harwitt's lab to identify it. So I could identify it, and when I asked them, are they really apoptotic, they said yes, for sure they are apoptotic cells. So we did use vital dyes that specifically stain apoptotic cells, therefore you are confident that you are doing the right stuff. Anyway, this is apoptotic pathway. So we discussed how this was originally discovered. So now in C. elegans probably this pathway just started sculpting organism shape, and if you failed to do it, it was still okay, the worm does not die. Like for example, SED3 homozygous mutants are viable, normal, no problem, with those few extra cells they are fine, but not so in your case, you would not be born. So this is how your head will look like. This wild type mouse, this is SED9 knockout, which is equivalent to SED3, okay, caspase 9, sorry. So caspase is a protease. This caspase 9 and SED4, wait, which one? Okay, this SED4 and APAF1, these are interchangeable. You can get rid of SED4 and put the human APAF1 in C. elegans, it will rescue the phenotype in C. elegans. So these are core conserved pathway over few hundred million years. So you will produce lot more neurons and they will stay there, your proper brain will not be formed, okay, and this digit separation will not happen, this will be webbed. So eventually embryo will be dead. So this is a cross section through its brain area, and you see this ventricle and the wall. Here the wall is thickened and you do not even see that space. Okay. So it is lethal in vertebrates, and this is the pathway in the 2 different contexts, and the orthologs are colored similarly. Like Eagle1 equivalent is this big bags, BCL2 is the SED9, and APAF1 is the SED4, and caspase 9 and 3 are the SED3 equivalent. Okay. So these are the C. elegans and this is the mammalian neurons, and mutations in this obviously will cause cancer as well. Like for example, cells that are not adhering to a surface where they should normally adhere, they are actually activated to commit suicide by apoptosis, and if that fails, if any of this fail in this pathway, then those cells will not die, instead they will become metastatic cancer. So there is a lot of cancer situations where the mutation is actually on genes involved in this pathway. So okay, so we are done with that. The next one that we are going to look at, the really lost signaling. So these are in addition to those 4, okay, only couple of them. One is apoptosis, very easy pathway, right, only you have SED9, SED4, SED3, that is all you need to remember. Eagle1 in between you have to bring in for that activation. So otherwise that pathway is straightforward. Actually none of them are complicated, all of them are easy to remember, you just need to look at it multiple times. So the next one we are going to look at is not paracrine. So far whatever we were seeing like the 4 main ones, they are paracrine. So the the ligand is secreted out and they bind to the receptor on cells at some distance, not very far away like the way endocrine system works. But the next one that we are going to see is called juxtacrine, where the ligand and receptor are membrane bound and therefore the cells have to be physically adjacent to each other. So the Drosophila names have stuck in the literature more popularly, and that's the receptor is Notch and the ligand is delta. Okay. So officially it is called Lin12 Notch signaling because Lin12 was the first notch like receptor discovered. So we already know, did we encounter Lin12? Yes, we did in vulval development. We will refresh our memory once more. Involved in vulva development. What does Lin12 do in vulva development? So when the primary fate cell wants to suppress primary fate on the two adjacent cells, yes, lateral signaling to make the secondary fate. Okay, in the context of lateral signaling is a generic description of that class. In that context it is to induce the secondary fate. So that is how in vulva development it is involved. It is required for fly eye development, retinal development in fly. In vertebrate nervous system, to tell a cell do not become neuron, instead you become glial cell, that is done by the notch signaling. And yeah, in optic neuron specifically, so that is where this is involved. So here the interaction leads to cleavage of the notch. So usually there are three cleavages. One, first of all, it does not exist as a single polypeptide, it is made as single polypeptide, but on its route to the membrane via ER and Golgi. In Golgi it gets cleaved and it becomes two polypeptides but attached to each other non-covalently, therefore it is like a heterodimer, and that is what is embedded in the membrane, that is S1 cleavage. Then the S2 cleavage happens extracellularly only upon delta interaction, and that cleavage triggers an intramembrane cleavage by a protein called presenilin, okay, or cell 12 in C. elegans, SEL12. So that presenilin cleavage releases the cytoplasmic domain, and that is the active notch, that is the transducer. That goes inside and associates with CS cell. I will not attempt to recollect all of them. L is lag1, C. elegans protein, so I know it. S and C are Drosophila names, I do not remember them right now. Okay. So that association with that transcription factor activates the downstream targets. So that is how notch delta transduces the signaling. So essentially here the exciting thing about this molecule is the receptor undergoes three proteolytic cleavages, one before going to the membrane, another one extracellular cleavage and another one intramembrane cleavage, then the active molecule translocates into the nucleus. Okay. So this is how it works. So the signaling is easy, but we have a complex model to explain how this finally contributes to development. Okay. So there we are going back to vulva for a short time. Vulva even little earlier than vulva also we will go to understand that. So we already know RTK pathway, the anchor cell through the RTK pathway is going to make this cell become primary fate. Okay. Now this is going to prevent the two adjacent cells, in every way they are equal in terms of their positioning. So they are called equivalence group, that is another keyword you need to remember. Equivalence group means cells that have equivalent potential in terms of developmental possibilities, okay, what fate they can assume. So these are equivalence group cells, and the Lin12 signaling between P6 P and P7P and P6P and P5P tells them not to become primary fate and they become the secondary. And the ones that are farther away, they do not get any of this news and therefore they go defaulted to become hypodermis because someone else is telling them do not become vulva. Remember that negative signaling by Lin 50. So as a result they become hypodermis. So now how this signaling happens has been understood before this developmental stage, at the stage of anchor cell formation. So there again the anchor cell and its sister, they both are equivalence group. They are called ZT1.AAA and ZT4.PPP. Do you understand what that means? It was the anterior daughter. Similarly in the other one, when ZT4 divides, posterior, great granddaughter of ZT4 on the posterior side. Okay. So those two cells are like this P7P and P5P, they are in identical situation. One of them can become, so that is illustrated here. Okay. So one of them is going to become ventral uterine precursor cell. You need to make the uterus, right. So the ventral part of it requires this cell, the sister of anchor cell, and the other one is going to become anchor cell. So either one can be either one of the two fates. Okay. Both are capable of becoming anchor cell, both are capable of becoming ventral uterine precursor. So the student and the mentor who figured out this, so they did lot of ablations, mosaic analysis to ensure which molecule is in which cell. Through all of this they figured out the ligand receptor are both produced by both the cells, and finally they put together this model that is totally by random chance. If one of them ends up producing little bit more ligand, okay, ends up telling the other one to produce more receptor and produce less ligand, and that is the initial difference between purely by chance. Then that initial difference is reinforced by feedback. So here you see both this is the signal, this is the receptor, both are producing both. If one is producing a little bit of more of the receptor and another one producing more of the ligand, this ligand producing cell is going to tell the receptor cell not to produce ligands, instead produce receptor, and that ends up in this difference. Then finally one becomes anchor cell, another becomes ventral uterine precursor. So this is the model proposed to explain how fate determination might work among equivalence group cells via the notch delta pathway in different contexts. This is probably how one cell becomes neuron, another becomes glial, if you want to think in vertebrate context. Okay. So this brings us to the end of the signaling pathways.